We're all quite familiar with the term introns. They are segments of DNA that are transcribed into RNA but are removed during splicing before making a protein. This is why we tend to call them non coding, as they do not directly code for amino acids. But what about the term introners? The 2025 article published by the University of California, Santa Cruz explores this newly discovered selfish DNA element. Introners are a type of transposable element or jumping gene that can move from one part of a genome to another. This links back to Gen 2052, where we studied how transposons can replicate and insert themselves into new DNA locations. Like replicative transposition, introners copy themselves, leaving repeat sequences behind, which can contribute to genetic diversity and increase protein diversity. They are considered selfish as this replication is done for their own survival rather than for the host. What's fascinating is that they can sometimes move between unrelated species, a process known as horizontal gene transfer. Similar DNA transposons, like spin elements, have also moved across very distant species, according to Pace et al. In 2008. This shows that genomes are not fixed. They can acquire new elements from outside sources, adding variation in ways I hadn't thought of. Scientists have stated that introners are one of the main ways that new introns appear with a species' DNA, and they can influence alternative splicing, allowing a single gene to produce multiple protein variants. Roy and Gilbert in 2005 also suggested that new introns can arise by processes different from those that created the first introns, which aligns with how introners may function. The study also highlights the scale and diversity of introners. Researchers found over 1,000 families of introners across the thousands of genomes they analyzed, with most of them appearing in algae, fungi, diverse single-celled organelles, and more complex organisms such as sea urchins. It could be that introners are a general mechanism for introducing new introns into genomes. As mentioned earlier, introners can jump between completely unrelated species. For example, one introner in the study moved from a sea sponge to a dinoflagellate, a single-celled marine protist. These two species last shared a common ancestor 1.6 billion years ago, so this transfer could not have happened through normal inheritance. Scientists hypothesize that introners might hitch a ride on giant viruses, essentially like a selfish element shuttling around another selfish element, to jump from one species to another. They believe that they will be able to observe more examples of horizontal gene transfer if they keep looking within the millions of eukaryotic species that exist. At first, I found it difficult to understand why selfish elements were of importance, but this article, alongside Gen 2052 content, has provided me with a better understanding of their broader significance. They help explain the origin of many introns, contribute to genome complexity, and impact alternative splicing and protein function. I specifically chose this article because it allowed me to connect my knowledge in transposable elements and genome variation to this relatively new discovery in genomics. Overall, this study broadened my understanding of genome evolution and transposable elements, expressing how diverse and adaptable genomes can be.